Thank you for the occasion of presenting to you my research project on the development of a preventive treatment for Alzheimer's disease. My research team is working on the development of cell and gene therapy for hereditary disease since 1987. We are mainly working on Duchenne muscular dystrophy, Friedreich ataxia, RYR1 mutation, and finally, familial form of Alzheimer's disease. Over the years, I have trained 69 master degree students, 27 PhD students, and 18 postdocs. Currently, my team has 12 people, including 3 research assistants, and one of my collaborators for the Alzheimer's disease project is Dr. Robert Laforce, who is a neurologist following patients with neurological disease. There is currently no effective treatment for Alzheimer's disease, and this disease is currently affecting over 6 million Americans, and this costs $593 billion per year to the U.S. economy. It is estimated that by 2050, there will be 12 million persons affected by Alzheimer's disease, and this will cost $1 trillion per year to the U.S. economy. I am proposing a completely new potential preventive treatment for familial form of Alzheimer's disease and for sporadic Alzheimer's disease. The proposed treatment is based on the addition of the amyloid precursor protein gene using the prime editing technology, which is derived from the CRISPR-Cas9 technology. The amyloid precursor protein is normally degraded by cut by the alpha secretase followed by a cut by the gamma secretase. This produces fragments of protein which are degraded without causing any problem. However, the amyloid precursor protein may also be cut by the beta secretase followed by a cut by the gamma secretase, and this produces beta amyloid peptides, which are 40 to 42 amino acid long, which are toxic for synaptic transmission and which are forming amyloid plaques, which are responsible for the death of neurons and thus for the memory problem. The treatment that I am proposing is based on an article that was published in Nature in 2012. This article demonstrated that a mutation in the amyloid precursor protein gene present in 0.1% of the Icelandic population can prevent the development of Alzheimer in these persons even when they are more than 95 years old. The mutation is the replacement of an alanine in position 673 by a threonine. My hypothesis is that the insertion of the Icelandic natural variant A673T with the prime editing technology will reduce the formation of A beta peptide, reduce the aggregation of the A beta peptide and the formation of amyloid plaque, and thus prevent Alzheimer's disease. My research team is currently using the prime editing technology to modify several genes, including the Duchenne muscular dystrophy gene, the RYR1 gene, which is responsible for malignant hyperthermia, and the NKX6-2 mutation, which is responsible for ataxia 8. In recent experiments using new modification of the prime editing technology, we were able to increase to up to 60% the percentage of gene edition obtained following a single treatment. So far, my research team has published 6 articles on the modification of the amyloid precursor protein, introducing the A673T mutation to prevent Alzheimer's disease. I personally own a patent on the reduction of the amyloid precursor peptide production via modification of the amyloid precursor protein gene using the CRISPR-Cas9 system, and a second patent is also pending. The short-term objective of my research project is to confirm that there is a reduction of the A-beta peptide production by inserting the A673T mutation in vitro using the prime editing technology. These experiments will be done in human neuroblastoma containing the London mutation, in human fibroblasts obtained from patients with the London mutation, in human neurons obtained by the differentiation of the patient fibroblasts containing the London mutation. And this will also be done in neurons of a mouse model containing the London mutation. These experiments in mice will be done in vitro and in vivo in this mouse model, and we will verify that the insertion of the A673T mutation prevents the memory problem in this mouse model. The long-term objectives are to develop a treatment of familial Alzheimer disease due to the London mutation and to develop a treatment for sporadic Alzheimer disease. We propose to do initial experiments in the London mouse model. We think this will take 1 to 2 years. The main problem being the delivery of the prime editing agents to the neurons in vivo. We will thus explore different delivery methods, the dual AAVs, extracellular vesicle, virus-like particle, lipid nanoparticle, and chitosan nanoparticle. Following these experiments in the mouse model, we will request the permission to do a phase 1 clinical trial in patients that have the London mutation. We think this will take 1 to 2 years. And then the very same treatment can be applied to prevent sporadic Alzheimer's disease.